Good day, students. Today's practical is titled Floral Morphology of Hibiscus rosa-sinensis. This hibiscus flower. And under this, we have some topic and flower of this floral formula and floral diagram. So we'll start with the flower. We're going to make a longitudinal section of this flower. This is the flower stalk or pedicel. We have the style, we have the anther, this yellow part, and we have this, the pole holding the anther is called filament. Then we have our stigma, we have our petals, we have our sepals and episepal. So let's go into dissection. So it's easier to cut it from the base. When you want to do flower morphology of hibiscus, you must have at least 2 flowers with you. One for your transverse section, another one for your longitudinal section. So we're using those for our longitudinal section. This will be for our transverse section. So now we want to cut it into 2 equal halves. Now, after cutting, every part of the flower will give you half of it. For example, you have out of this 5 stigma, we have 3, at least 2.5. You can see our flowers, we have 2.5. Now, let's draw this flower. We are still on half flower. When we want to come to floral formula, we'll talk more on what we're doing. This is half flower. Every part that passes through, to cut it into half, you represent with a double line. For example, we have this was not cut, this is cut, and this is half. So this half will represent the double line. In biology, anytime you cut a part of a plant into half, you represent that half with double line. Now let's go to the board. So we have our this will represent the double line because it's half. So when you are drawing, you make sure you have a free hand drawing, so you don't need to be cleaning. So we have our flower. We have our flower. We have our flower. Now you can see our ovary. We have our receptacle. Here we have our so this is our flower stalk. This is our episepa. This is our sepa. We have our ovary. So this is our ovary. This is our ovule. This is our pedicel. This is our petal. Style. Now, in this place, we have our flowers. So this is our anther. This is our filament. And this is our stigma. So, because we have cut this part with blade, to represent it with double line. Because all these parts have been cut with blade, we represent them with double line. All the parts that are cut with blade, represent them with double line. Every part you cut with blade, represent them with double line. Double line. This part was also cut with blade, represent with double line. So here we have our half flower of hibiscus, rosa-sinensis. Sepa. So this is half flower. This is the first part of our experiment. Half flower, we have sepa, flower stalk, and so on. So thank you. This is the first part of our flower. Now we are done with half flower. We want to do floral formula for our hibiscus flower. Now this floral formula will be much more easier if we have, for example, now this plant can be, we have to look at the symmetry of the plant. Can it be cut into 2 equal halves? From this, we have 5 of the petals. Yes. It can be cut from every plane to 2 equal halves. So the first, our formula will be axillomorphic, that is radially symmetrical. You can cut it to any plane. Now, is it both male and female? You can see the male part, the anther, and the ovary. You can see the ovary represent the female part. So it is an hermaphrodite flower. Then let's look at from this side. This is epicalyx. How many of these do we have? 1, 2, 3, 4, 5. We have 7. And these 7, they are free. They are not joined together. That's why you can see the 7 of them. Epicalyx, they are free. They are not joined together. You can see they are not joined together. Now, the next one is calyx. This is calyx. That is the sepal. Calyx are a group of sepals. So this is calyx. You can see that we have 1, 2, 3, 4, 5. There are 5. They are joined together like a cup at the base. So they are fused together. So our calyx is 5, but they are fused together. Then the next one is the androecium, the male part. You can see this yellow part that has the filament carrying the anther. They are more than 15. So our androecium, which is the male part, is A is infinity, is above 15. Then we now look at our gynoecium. Now, how do you get your gynoecium? You have to open the flower. That's why I said you need 2 flowers to do this experiment. You can save our ovary. After opening, this is our ovary. This our ovary is a superior ovary. You can see it is above the receptacle. Our ovary is, this our ovary is a superior ovary. So what we do is we dissect the ovary. We do transverse section of the ovary this way. Then we get a very small section and place on the microscope slide and examine under the microscope. If you check the previous video, this has been done and our carpel number is 5. So G is 5. Then our ovary is superior because it's above the receptacle. So you put your bar below. So this is the floral formula of Hibiscus rosa-sinensis. This is the floral formula. Now let's go into the floral diagram. We've seen it is radially symmetrical, actinomorphic, it is hermaphrodite, 7 epicalix. We've not seen one part is there. We've not done the whole part. We've not checked the corolla. Now, after the androecium, after the calyx, we have the corolla. Let's go back to this. Calyx, we have corolla. How many corolla do we have? 1, 2, 3, 4, 5. Corolla are groups of petals are called corolla. So we have 5 corolla. And because they also join at the base, we said they are fused together. Then we're now going to androecium, where we have more than 15. Is infinity. Then our gynoecium, the ovary is superior. You can see the ovary is above. And then when you do transverse section of the ovary, you get, like we've done in previous video, our gynoecium is 5. Our carpel number is 5. So, and because the ovary is superior, is above the receptacle, is superior, it's very obvious. So we have these are floral. So we have actinomorphic, bisexual, 7 epicalix, 5 calyx fused, 5 corolla fused, infinity androecium, and 5 carpel. Thank you. Let's go to the third one now.